Dear student, today we discuss about Schistosoma heamatobium. First of all, we looking to see classification of this organism. Phylum Platyhelminthes, class Trematoda, order Digenea, genus Schistosoma, species Heamatobium. Generally, the Schistosoma is also called Bilharzia. Generally, this is a parasite discovered by Bilharzia. That is the reason why we are calling as a Bilharzia. After that, this organism which lives in the blood, it is also called blood fluke. And the male has a special cavity, cysto. So that is the reason why this species treated as a Schistosoma. There are main three species in a human being infections and causes diseases: Schistosoma heamatobium, Schistosoma japonicum, Schistosoma mansoni. These are the main three species which causes the disease, and some other organisms also causes different diseases like birds and some mammals. So in birds, Cercarial dermatitis takes place, and in human beings also Cercaria cause some dermatitis. And the some Schistosoma mansoni and heamatobium most inhabit vascular and pelvic venous plexus. Generally, these parasite which lives at the region of the pelvic region, especially where the pelvic region blood vessels are present. Those in that blood vessels, these organisms can inhabit it. Okay, so now this is generally the the other organisms also hosts. Generally, some different hosts are there. The various animals such as cattle, dogs, and cats, rodents, pigs, and horse and goats serve as a reservoir. Okay, so now this is digenetic parasite. Primary host is man. Secondary host is a snail. Bullnus planarbis, Bullnus truncatus. Okay, so some hosts are there, and some reservoirs also there. Next, geographical distribution. Schistosoma monsoon is found primarily in sub-Sahara Africa and some South American countries like Brazil, Venezuela, and Suriname, and the Caribbean with sporadic reports in Arabian Peninsula. Generally, especially there is a parasite which found in sub-Sahara, especially African countries. Okay, the Schistosoma hematoma found in Africa and packets of the Middle East, and Schistosoma japonicum, which found in China, Philippines, and mm, some other Japan. Okay, next one. Uh, so the other less common human infecting species, especially Schistosoma macogni, occurs finally parts of the Cambodia and Laos. So some different parasites are there. Next, structure of the Schistosoma heamatobium. Generally, the Schistosoma heamatobium is it can show remarkable sexual dimorphism, and the male is thick and smaller than a female, and the male size is up to 50 mm, and the female size is up to 20 mm. So generally, the male has a thick, the female has a thin, and generally, the structure of these Schistosoma heamatobium. So anterior side, some oral sucker is there, and below oral sucker, esophagus is there. Below the esophagus, some acetabular or a ventral sucker is there. So below the ventral sucker, the male has special cavity, so that is called gynecophoric canal. So in this canal, the female is present. In this canal, the female is present. So always looks like in reproductive condition. So that is the reason this is called a wedded condition. And in the female generally inhabited in the male's cavity. That cavity is called gynecophoric canal, and generally the male has a number of rhabdite like spinules like structures which is present on the surface of the male, and the female is very thin and length in large. So this is about structure. And next, so we have to see diagrammically. So generally, This is real worm structure, and the oral and ventral sucker of male worm. So generally this is oral and ventral sucker, and the gynecophoric canal is there, and structure of the body, so head and tail, and further cancelled in gynecophoric canal. So generally the female is present in gynecophoric canal. Now, reproductive system. First of all, male reproductive system. Generally, 4 to 7 testes are present in the male reproductive system. 4 to 7 testes are present in the male reproductive system, and these testes are the site for production of sperm. Generally, this testis has germinal epithelium. So, this germinal epithelium undergoes reduction division or meiotic division. So, these cells can produce the sperm. Okay, the testis directly opens into vasa efferens. Vasa efferens is a small tube. Now, the union of number of vasa efferents, they can form a vasa deferens. Okay, and now the vasa difference is large in size, that is called bulbourethra, and finally these genital aperture opens near to the ventral sucker, near to the ventral sucker. So this is about male reproductive system. Here the testis, vasa efferens and vasa deferens, and the pore, genital pore. So this is about male reproductive system. And next we are going to female reproductive system. Only one ovary is present in Schistosoma heamatobium. One ovary is present. Generally one ovary directly opened into nearly uh, vitelline duct or oviduct. So near here, the where the oviduct is present, the yolk glands are present. Yolk glands. So these yolk glands are useful for to producing the capsule around the ovary, around the ova, and along with some yolk cells. Generally, these yolk glands can secrete the capsule, and along with some yolk cells around the ova, so they can form a capsule. So in the capsule ova is present, some uh, yolk cells are present. Generally these yolk cells are give the nourish to the zygote whenever the embryogenesis is takes place. Whenever the embryogenesis is takes place, here the yolk glands are present, and now the vitelline duct and oviduct move forward, and it reaches to the nearly ventral sucker, and the two vitelline ducts are unite to form a oviduct. So here it is the O type, and around the O type some Mallis glands are there. Mallis glands. So these gland secretion is useful for the movement of the capsules, and these oviducts opens into uteri. Uteri directly opens through the female genital pore near to the ventral sucker. Near to the ventral sucker. So here we have to keep it in mind. One ovary is there, two oviducts are there, and common oviduct and uteri and female genital pore. So in female one ovary is there. In male 4 to 7 testes are there. So generally the ovary cells, the ovary has genital epithelium. So these epithelium undergoes reduction division or meiotic division. So the ovas are produced along with some polar bodies. Along with some polar bodies. Okay, so this is about male and female reproductive system in Schistosoma heamatobium. Male and female reproductive system, Schistosoma heamatobium. Next one are your life cycle. Generally, these Schistosoma heamatobium, which lives in the pelvic region of the human beings, especially near to the uh, um urinary bladder, near to the colon, and near to the large intestine. Okay, especially three types of parasites we can observe. So Schistosoma heamatobium, Schistosoma japanicum, Schistosoma mckenzie, Schistosoma mansonium. So generally these species has some different structures of the capsule, different structures of the capsule. So the each disease can producing the peculiar size of the capsules, peculiar size of the capsule. So whenever generally these in this organisms life cycle takes place in two hosts. Primary host is man, that is Homo sapien. Secondary host is snail, that is Bullnus truncatus, Bullnus planarbis, Bullnus truncatus, Bullnus planarbis. So nearly number of species are there mm, as a secondary host for these Schistosoma heamatobium. Okay, so generally the reproduction takes place in these blood vessels of the pelvic region of the human being. Now the male genital pole release the sperms into female genital pore. Okay, so the fertilization is takes place. By the fertilization, zygote is formed. So by the male glands and by the uh, what is yolk glands. So these yolk glands can secrete the some cell and some capsule around the yolk cells and zygote, so they can form a capsule. Okay, so this capsule has a spine, and this by this spine, so these eggs are released into the urinary bladder, and sometimes the eggs are released into the large intestine, or sometimes eggs are released into the small intestine. So whenever these eggs release into the bladder or urethra, these eggs are come out of the urine through external environment, and the capsules released into the large intestine and small intestine. These capsules are come out of the fecal matter. So whenever these capsules released into the nearby the legs, so whenever these eggs are attaches with water or touch with water, immediately the miracidium larva are released. The miracidium larva are released. Generally, the inside the capsule, already the miracidium larva was developed. Miracidium larva was developed. So this larva has especially penetrating glands to penetrate into the snail body, into the liver of the snail, and some flame cells also present, some intestinal cells also present. So these miracidia has cilia for movement. So the miracidium freely swimming in the water, and the miracidium live up to survive up to 24 hours. After 24 hours, the miracidium may not reaches the snail, it may die. Okay, so now whenever the miracidium reaches into the snail, and by the penetrating gland, the miracidium enters into the snail liver, and here the immediately the sporocyst is formed. The sporocyst develop in a snail successive generation. Generally, here the polyembryogeny is present. Polyembryogeny. One larva can generate next generation larva. So one larva can producing next generation larva. This phenomena is called polyembryogeny. Polyembryogeny. From one miracidium, number of porocysts are developed. So in Schistosoma heamatobium life cycle, there is no formation of redia. In Fasciola hepatica, redia larva is also present. Now from the sporocyst, the sporocyst has some intestinal cell. By these intestinal cell, these cells can produce next generation larva, that is Cercaria. This is free swimming larva. So the free swimming larva has forked tail. By the tail, so the Cercaria can swim. So generally the Cercaria larvas are producing in the snail, and these larva has especially penetrating gland, and ventral sucker, and digestive parts, and some cystogenous glands also present. But the cystogenous glands are not useful in Schistosoma heamatobium. These glands are useful for Fasciola hepatica. So now these Cercaria larva, which released into the water from the snail, and whenever the human beings are consuming this water by these Cercaria larva, generally these Cercaria enters into the human being. And sometimes whenever the human beings are entering into the water body, place the legs into the water body, so the Cercaria larva penetrates into human being through these penetrate glands. Okay, so the infection takes place either consuming the water, or infection takes place swimming in the water through skin, or place when we are place the water into the human water body. Generally these Cercaria larva penetrates into the human beings. So the Cercaria larva loses the tail, and it can form as a schistosomula. It can form as a schistosomula. So this schistosomula enters the circulation, and it can reach us to the heart, lungs, and liver, and finally it migrates to the portal blood vessel in liver, and maturation to adults. So these organisms are maturate into adults, especially in portal blood vessels, and finally it reaches to the pelvic blood vessels. So whenever it reaches to the pelvic blood vessel, so it fully developed, and the male is attached whenever recognize the female, and its gynecophoric canal engulf the are surrounded by these female, and generally whenever the male identify the female, the gynecophoric canal is formed. So in the gynecophoric canal the female attached. So the Schistosoma heamatobium always in reproductive condition or sexual condition. So this condition is called wedded condition, means wedding. Okay, so this is the life cycle of Schistosoma heamatobium. So Schistosoma heamatobium, which lives in the pelvic blood vessels of the human being. So here the reproduction takes place, the zygote is formed. From the zygote, capsules are formed by the male glands and uterine glands. So now the capsules are released into the either urinary bladder or large intestine and small intestine. So these ovas are come out of the pores by excretion, or either urine or fecal matter. So whenever these eggs are reaches to the water body, and immediately whenever these eggs are touched with water, the miracidium larva is released. Now the miracidium larva is a free swimming larva. It can swim and identify the snail, that is Bullnus planarbis, Bullnus truncatus. So by its penetrating gland, the miracidium enters into the snail. Okay, now whenever it enters into the snail, it will become a sporocyst. Here the radii is absent, and from the sporocyst the Cercaria larva is formed. Again Cercaria larva is infection stage to the human being. Okay, so here the infection stage of snail is, that is miracidium, and infection stage of the human being is, that is called Cercaria larva. So this is about life cycle. And next one is clinical presentations. So symptoms of schistosomia are not caused by the worm. Generally the body reactions to the eggs. Whenever the eggs are released, so immediately the infection takes place, especially the local cutaneous hypersensitivity reaction followed by the skin penetrating the Cercaria. Whenever the Cercaria larva penetrates into through skin into our body, so hypersensitivity reaction takes place, nothing but allergy. So our WBC can release the antibodies like IgE, that leads to allergy. That allergy in another word is hypersensitivity. Okay, generally the allergy takes place in the skin, and some itch, so some catamaya fever, so hypersensitivity reactions, different diseases. So now especially these eggs are released into the bloodstream. So generally that leads to fever, cough, abdominal pain, and diarrhea, and hepatosplenomegaly. Generally the spleen size is increased, and the eosinophils number is also increased. So in one eosinophils are one type of blood vessels, so these eosinophil number is increased. Eosinophil number is increased, and the different cystosoma has a different characteristics. Some organisms cysts are entering into the central nervous system, so that leads to cerebral granulomatous disease, and some cystosoma japanicum eggs released into the brain, and some brain lesions takes place. Okay, so these are the different cystosoma heamatobium activities or effects on human beings. So how to identify? So we can identify the Schistosoma heamatobium by the urine examination. So number of capsules are we can identify by the fecal examination. We can identify number of fecal matter capsules of Schistosoma heamatobium in fecal matter. So next treatment. So we can control the disease with praziquantel drug. So all Schistosoma heamatobium stages can control by the praziquantel drug. Okay. Next one is prophylaxis. So how we can control? So human fecal not pollute with drinking water. So generally we have to use sanitization, proper institutional sanitation and personal sanitation. So we have to use mm, bathrooms and toilets, and generally there is no fecal matter contaminated with drinking water, and kill the snails. The snail is uh, secondary host, so we have to kill the snails. And another thing, stop swimming, drinking, bathing infected water. Generally in rural areas like poor countries, or in that poor countries there is no proper sanitization. So the most peoples are they can complete their activities, fresh up activities near to the lakes. So whenever the infected persons are release the urine and fecal matter near to the water bodies, so the chances are there these eggs are entering into the water body. So immediately that contaminated water drinking by the human beings, or whenever the persons in the contaminated water human beings are perform swimming or practicing the refresh of cloth. So this is the time the Cercaria larva enters into human being. Okay. So this is about Schistosoma heamatobium life cycle. So here we have to remember it, the life cycle. And the in human being pelvic region, generally the pelvic region blood vessels, in pelvic region blood vessels Schistosoma heamatobium survive. And here, so these females are released the capsules, and capsules are entering into the water body. So immediately the eggs release the merothelium, and merothelium penetrate into snail after before 24 hours, and the merothelium convert into what is sporocyst. From the sporocyst the Cercaria larva is present. Cercaria larva is next contaminate stage to human being. Here the polyembryogeny takes place. One larva can producing next generation larva. This phenomena is called polyembryogeny. So here these platyhelminthes organisms life cycles are complex life cycles we can identify. Complex life cycles we can identify. And male female reproductive system. Male reproductive system has 4 to 7 testes, and female reproductive system has only one ova. In female reproductive system, what is their male is gland and yolk glands are present. Yolk glands can producing the yolk and producing the yolk cell, and male glands are useful for to transport the eggs into uteri. So this is a real worm structure. This is a structure of the Schistosoma heamatobium. So this is a life cycle of Schistosoma heamatobium. Shall we start?